We're all familiar with grudges and the harm that they can do to our relationships. But we don't often think about the grudge that our immune system holds against every bacterial and viral infection that we've had in our life. An important component of these grudges come in the form of memory T cells. After an infection, these memory cells are formed and they patrol our bodies. That makes these cells a very important component of our health, and understanding the factors that regulate them has been a key question in the field of immunology. So far, most of this work has focused on genes that make proteins, which we call coding. However, there are many genes that do not make any protein at all and only make RNA, which we call non-coding. What's really exciting about my work is that we discovered how one of these non-coding genes, called Malat1, acts like a sponge to regulate these important memory cells. Traditionally, these non-coding genes have been harder to study than their protein-coding cousins. So to address this problem, we turned to cutting-edge DNA editing technology to create a new mouse line. In this mouse line, we were able to edit Malat1 specifically to remove its sponge function, but leave all its other functions and expression levels intact and untouched. We could then understand how these spongeless Malat1 mice respond to bacteria or viral infections. When we infected these mice with a bacteria or virus, we found that the spongeless cells weren't activated as well and produced less immune stimulatory proteins that helped to destroy the invader. Further, after the infection was resolved, we found that there were less of these important memory cells in these Malat1 spongeless mice. This will allow us and other immunologists to have a better understanding of all the factors and consequences of regulating these important memory cells. But this can also have implications for future clinical trials in humans. One thing that we've learned from the COVID-19 mRNA vaccines is that it's relatively safe and easy to administer RNA, like Malat1, to our body cells. So then we can imagine introducing more Malat1 into these memory cells to boost their function. This could lead to better development of vaccines and cancer therapies. So being able to manipulate Malat1 may just be a way for our immune system to hold onto those grudges a little while longer. Thanks to everyone who made this work possible, and thank you for listening.